Hello everyone, welcome to my presentation. Our topic for today is all about the Bacteroides fragilis bacteria. And this bacteria can be found anywhere in our places surrounding us. To know me, my name is Charity Trozo Sabatin from BSA 3B of Northeastern Mindanao State University. I know that some of you are not familiar with this bacteria and others are also familiar. So if you wanted to know and if you wanted to learn about this, keep watching and I hope that you will learn. First objectives: determine the cause of Bacteroides fragilis infections and outbreaks. Second, examine the proper diagnosis of Bacteroides fragilis infections. Third, explain some interprofessional team tactics for advancing the management of Bacteroides fragilis infections and improving outcomes by increasing care coordination and communication. What is Bacteroides fragilis? Bacteroides fragilis is a part of normal microbiota of the human colon and is generally commensal, but it can cause infections if displaced into the bloodstream or surrounding tissue following surgery, disease, or trauma. Gram-negative bacteria, non-motile, anaerobic blood agar colonies are non-hemolytic and gray with an entire margin and ring-like structures. It is saccharolytic, which means extremely virulent and can cause widespread tissue destruction, has a capsule and also produces several enzymes, no pigment or spore formation. In the 1980s, the genus Bacteroides comprised a large and heterogeneous group of anaerobic gram-negative bacilli, but over the past 3 decades, many species formerly assigned to this genus have been reassigned to other genera. And this is how it looks like the Bacteroides fragilis. Where we can find its habitat? Well, Bacteroides fragilis resides in the human gastrointestinal tract and is essential to health gastrointestinal functions such as mucosal immunity and host nutrition. As a mesophile, optimal growth occurs at 37 degrees Celsius and a pH around 7. It has causing disease in humans such as human colon is lined by a mucosal barrier that protects body tissues from being invaded by the bacteria that inhabits the intestinal cavity. Fragilis is a seniculture with a mix of bacteria. 3 different liquid overlays were evaluated, including Ehrlich's balanced salt solution or EBSS, PBS, and Dolby-Co's modified PBS for their ability to support the in vitro growth of the Fragilis trophozoites. This picture shows how the strain competitions restricts colonizations of an enteric pathogen and prevents colitis. Enterotoxigenic Bacteroides fragilis or ETBF cause acute diarrhea and are associated with active inflammatory bowel disease or IBD, late-stage colon cancer, and sepsis through production of B. fragilis toxin or BFT. Conversely, non-toxigenic B. fragilis or NTBF strains are symbiotic, protecting their host against inflammatory disease. The microbiota is a major source of protection against intestinal pathogens. Our studies demonstrate a novel role of type 6 secretions systems and colonization resistance against a pathogen. This understanding of bacterial competition may be used to the microbiota is a major source of protection against intestinal pathogens. Our studies demonstrate a novel role of type 6 secretion systems in colonization resistance against a pathogen. This understanding and bacterial competition may be used to define a molecularly targeted probiotic strategy. What does it mean if your Bacteroides fragilis results too high? Gram-negative species of the bacterium of What does it mean if your Bacteroides fragilis results is too high? Gram-negative species of the Bacteroides phylum, immune-modulating normal gut species believed to be involved in microbial balance, barrier integrity, and your neuroimmune health. High levels may result from reduced digestive capacity or constipation. Bacteroides fragilis is a part of the normal microbiota of the human body. Immune system begin to combat the infection. Another study identified that Bacteroides fragilis species have a protective effect against antibiotic-associated diarrhea in rats by alternating intestinal defenses and identified the vital role of B. fragilis. How is Bacteroides fragilis transmitted? Mode of transmissions: infection results from displacement of Bacteroides spp or closely related genera from normal mucosal. Let's talk about their treatment and management. Cefoxitin, mosifloxacin, and clindamycin have low levels of susceptibility for Bacteroides fragilis, whereas piperacillin, tazobactam, meropenem, and metronidazole have high susceptibility rates. Metronidazole is the antibiotic of choice for the management of infections caused by anaerobes. End of my discussion. I hope that you learn. And that's the end of my discussion. I hope that you learn more about this bacteria named Bacteroides fragilis. Once again, this is Charity Trozo Sabatin from Northeastern Mindanao State University. Good night and God bless.